Okay, we're on.
All right, let's look at phosphorylation. Phosphorylation is a covalent attachment of a phosphate ion, a negatively charged phosphate ion, to a particular part of a protein. Now, in order to do phosphorylation, to attach that phosphate to the protein, that's a condensation reaction. It takes energy. We also have to have a source of phosphate ions to do this. And what we're going to do is a coupled reaction that provides us with both. So we're going to have a protein here, and an enzyme binds to it, and it also binds to an ATP. The ATP is going to be the source of the energy and the source of the phosphate. That enzyme, we'll see what these are in a moment, is going to tear the third phosphate off of the ATP, which is highly exergonic, and then do the condensation reaction, attaching that phosphate to the protein. So now we have, with a covalently attached phosphate, I'll just represent as a P with a circle, and we release ADP. Okay, now the enzymes that perform that are known as the kinases, more specifically the protein kinases. Okay, so that's the forward reaction. That's a condensation reaction. Now, when we want to do the reverse, get rid of that phosphate, we call that dephosphorylation. And we'll start here with our protein that's phosphorylate, has the covalently attached phosphate. Okay, an enzyme performs a hydrolysis reaction that breaks the phosphate off of the protein. So now we have the protein again. And the phosphate ions released free into solution. Now, since that's a hydrolysis reaction that does not require energy, it's an exergonic reaction. However, we still need enzymes to catalyze that, and these are called the protein phosphatases. Okay, so once again, we need a certain set of enzymes to do the phosphorylation. That's the kinases. And then the phosphatases are a second set of enzymes that do the reverse, that dephosphorylated protein. So that's what we see with our forward and reverse reaction. By the way, common test question too. Fair warning.
And the second one's the energonic.
No, the second one's exergonic because it's hydraulic. The first one's endergonic. That's why you have to have the ATP, not only phosphate, also the energy. Question? All right. All right. Now, what I'm going to say here applies to phosphorylation and dephosphorylation. We're going to make several points here. This will also apply to acetylation as well, which we'll look at much more quickly. Point number one, there is no such thing as a general one-size-fits-all kinase or phosphatase. Each kinase and each phosphatase has specific protein targets. It will not touch other proteins because the appropriate part of the protein cannot fit into the active site of the kinase or phosphatase. Some kinases and phosphatases regulate many different proteins. Others regulate just one and nothing else. So every single kinase, every single phosphatase has specific target proteins. What that means is the majority of our proteins are regulated, at least in part, by kinases and phosphatases. Now that means you're dealing with tens of thousands, possibly hundreds of thousands of regulated proteins this way. What that means is you have to have lots of different kinds of kinases and lots of different kinds of phosphatases around. In the human genome, with about 22,000 genes, you add up the total number of genes for kinases and phosphatases, and that exceeds 1,000. That's about 5% of our total protein coding genes. So it tells you they're important. That's a lot of your genes that go into these guys. Okay, second thing. Kinases and phosphatases do not stick phosphates or remove them randomly, even on their target proteins. Each kinase, each phosphatase targets only one specific location on that particular protein. An amino acid, the number 50 amino acid, number 300 amino acid, or what have you. So each kinase and phosphatase So each kinase and each phosphatase targets only a specific amino acid on the target protein where it sticks the phosphate or removes it from. As a matter of fact, only certain amino acids on a protein can get phosphorylated. Most kinases and phosphatases target the amino acids called serine and another one called threonine. Those are the common targets of most kinases. But there are some that can phosphorylate the amino acid tyrosine. And when these so-called tyrosine protein kinases were first discovered, it generated a tremendous amount of interest because the very first tyrosine protein kinase was coded for by a gene in a cancer-causing virus that affects chickens and other birds. And they identified this one gene that could cause cancer in these birds when the virus infected them, and they found it was something totally unusual, a protein kinase that phosphorylated tyrosines. This was back in like the 1970s or so. Not surprisingly, there was a huge amount of research looking for tyrosine protein kinases. And we find that many of them are indeed involved, at least when they're misactivated, misregulated, many of them are indeed involved in the biology of cancers. There's a lot of interest in these guys because they play roles, and when they go wrong, they can affect and stimulate cells towards a cancerous state. So that's another kind of thing. Each kinase and phosphatase has specific targets. A third point I'd like to make here . Okay, a given protein can be regulated by many different kinases and phosphatases at many different sites. So in other words, a particular protein may have several different sites that can be phosphorylated or dephosphorylated in different locations on the protein. Each one of those events is going to have some kind of effect on the protein. And likewise, that means that a given protein can be regulated by multiple kinases and multiple phosphatases, each with its own specific target, and each with its own specific effect. Now, one other thing I like to mention is kinases and phosphatases themselves are regulated proteins. Many times they can be regulated by phosphorylation, or they can be regulated by other means. So